Yes.
Okay, wonderful. As I mentioned, it's wonderful to speak with bright undergraduate students, and I teach undergraduate students as well. So it's wonderful to be here. You see the picture there of Reed College on a much sunnier day, actually in daylight instead of darkness. And my talk today is going to be about how we have discovered these bacteria that degrade plastics, as Mansi said. And the connection is that I have studied infectious disease for the beginning portion of my career, but there are a lot of similarities in terms of genetics and interaction with the environment, which allows us to study these bacteria. I really have two parts of my talk today. The first part is going to show you the evidence for the bacteria being able to degrade the plastic, and I will tell you the story of how we discovered the bacteria, and then the evidence for how that's happening, and the ongoing research, which is funded by the National Science Foundation and engaging undergraduate students. The second part of the talk is going to be a more practical aspect of it, where I'm going to show you some ideas about how we might use this technology and how to improve it to be able to recycle PET plastic or water bottles, which you know is an immense problem. And you've done a lot of work with this as well. And all of that data is unpublished. So I'm going to go ahead and move forward here and start with a little bit of the problem, which, as I mentioned, most people are very aware of, particularly in the pandemic when we have more time to think about things. So plastic is a blessing and a curse. It's very durable. We've used plastic materials really widely since about the 1950s. Nobody really knows exactly how much plastic is in the environment, but we have some pretty good estimates. So I have some chemical structures of some of the common plastics in the middle portion of the screen down here. Can you see my pointer?
Yes,
yes, we can.
Okay, wonderful. So this is PET plastic, of course, the material used to make water bottles, but there's also polyethylene, polypropylene, polystyrene, and so forth. A lot of different structures which are difficult for bacteria to degrade because they've not seen these before out in the environment. They're beginning to evolve to degrade plastics, but the process is still very, very slow. And there's billions, hundreds of billions of PET water bottles that are produced globally every year. And we have an ongoing and increasing problem. Many of you, I'm sure, have seen pictures like this or been in environments where you see plastic waste and your heart sinks, particularly, I'm sure, from being in a zoology department to see that. This estimate is actually fairly low. There's over 300 million metric tons of plastic waste that are generated every year. And polypropylene, polyesters, and PET plastic are certainly part of that. The US produces about 10% of that waste. A lot of water bottles and other single-use containers. One of the problems is that it's difficult to recycle these. And only 10% or 9% in an estimate, recent estimate of any sort of plastic has ever been recycled. 6 billion tons that currently exist in the environment. That's the huge burden for an environment. So some more recent information, I'll try to move on to the more optimistic information, but just to let you know sort of the gravity of the problem. There was a publication in Science in 2015, Jambeck et al., that made some predictions in terms of the amount of plastic in our oceans versus fish. And they predict that by 2050 on the current course, that there's going to be the same amount of fish in the oceans as plastics, which is kind of a horrific thing to think about. And it's also using a lot of the petroleum products or oil consumption in the world predicted now to be about 6%. And if we continue on this track to 2050, to be about 20% of oil, of petroleum products. So we'd like to get away from that. We'd like to not only clean up the environment, to be able to recycle more efficiently, and also not use fossil fuels to be able to produce these materials because there's so much of them already. So here is some information that I really didn't know until I got started with this project. And that is that it is really difficult to recycle. And I didn't really understand why. So less than 10% has been recycled. All of us put our plastics in recycling bins. We feel good about it. But really, the reality of the matter is that very little of that material actually does get recycled. Excuse me. If they do get recycled, often PET plastic is just turned into clothing or carpet, and then it goes into the landfill after that. It's not a circular economy or circular process. And the reason really, which I didn't understand before, is really about the plasticizers. So those are the materials or the chemicals that manufacturers place into the plastics to give them the characteristics that they're interested in, to be able to put carbonated beverages or water in containers and not have them leak. But those plasticizers or molecules make it really difficult to purify the components, which I'll tell you about, that are necessary to make new water bottles. So recycling is really challenging for that reason. There is some effort to be able to biorecycle. So using enzymes, for example, that occur in bacteria, mostly soil bacteria or bacteria that have been isolated from waste sites. And so you probably have read about the bacterium that the Japanese group have isolated that are able to degrade PET plastics, or the other engineered cutinases and other such molecules. All these enzymes, as I'll tell you, are in the category of alpha beta hydrolases that cleave ester bonds. But there's a lot of work in this, but also it's quite expensive to be able to isolate enough enzyme in the laboratory to put on millions or billions of plastic water bottles to be able to degrade them. So that's going to be quite an effort to do that. So our approach has been different. Reduce, reuse, recycle, biodegradation. What we would like to do is to be able to use the whole organism, because bacteria are easy to grow and fairly cheap, to be able to then degrade PET plastics and then to be able to recycle them. So a few years ago now, my undergraduate student, Morgan Vague, whose picture is there on the screen, she's from Houston, Texas. And so there's a lot of petroleum industry there. There's a lot of pollution in Galveston Bay, which is pictured on the left there. And so she came to me, she took my microbiology class, and she said, "Hey, Jay, I read about this Japanese group that isolated this bacterium that degrades plastic. Can we do that?" I said, "Heck yeah, we can do that." And so she went home for the semester break, and she collected some soil and put it in her luggage, and she flew back to Portland. And then we started to work on these bacteria. And so she screened for lipase activity, or the ability to cleave lipids. So if you cleave the fatty acid chain off of a triglyceride and combine it with this dye called rhodamine B, which is here listed, and in this agar, and you shine UV light on it, you get fluorescence. And so it's a really easy way to what we do to screen or to look for these bacteria that are able to then cleave this type of molecule. So then what we did was we began to characterize them a little bit more carefully. And the substrate that we used in these plates was just simply olive oil. And then when those fatty acids get cleaved, then they combine with the dye, and you can shine UV light on them and you can see the halo. And so you'll notice then, I'll tell you about these 5 bacterial strains, but basically these strains here, these were Pseudomonas species. You'll notice the numbers 9.2, 10, and 13.2. And also one thing that's confusing is they naturally fluoresce. So you can see in the absence of UV, or the absence of the rhodamine B, you still get some fluorescence here. So that was a little bit of a confounding factor, but we worked it out. And so ultimately what we did was we were able to identify these consortia of bacteria. And that's a whole different seminar in itself, because these 2 bacterial species, Pseudomonas and Pseudomonas and Bacillus really do work well together. So there's been other instances, which I'll share a little bit with you later on, other instances of Bacillus species and Pseudomonas, which are common soil bacteria, to grow, live together. And so what Morgan was able to do was identify these two consortium, containing Bacillus and Pseudomonas, Consortium 9 and Consortium 13, and a single isolate of Pseudomonas 10, isolate 10, which she called it. And so the bacteria that were lipase positive were gram negative, which are stained pink here. And the lipase negative bacteria were stained purple.
Kindly mute yourselves, please. Sorry for the disturbance.
Oh, that's okay. You got to be flexible when you're on Zoom. So, okay, anyway, so she isolated these 5 bacteria, and 3 were Pseudomonas species. They were gram negative, which appear to be pink here, which were lipase positive, and the Bacillus species, which are purple, were lipase negative. They grew together. And then we published the draft genomes in a microbiology resource announcement, which allowed us to then identify some of the genes that are involved in the processes. So I want to tell you a little bit about the methodologies that I'll share with you today, which are pretty broad. These methodologies are used to, as I mentioned, be able to understand or to be able to quantify the degradation of the plastic by the bacteria. So we're using microbial culture, simply growing them. We can weigh the plastic before and after. We're using biochemical and biophysical means, as well as genetic tools, as I shared with you, to be able to characterize the plastic. Another talented undergraduate who worked on this project is Cam Roberts. She's pictured down there on the left, and she did her thesis. So at Reed College, we have a required senior thesis. And so she was able to show, just by simply growing the bacteria on plastic, that the consortium grew better than the single isolates. And so you noticed in the graphs here, we're just plotting the absorbance or the optical densities at 600 nanometers of the culture, which is common in bacteriological studies. And so you can see here that this consortium containing all 5 isolates grew more rapidly and to a higher yield than any of the individual consortia or the isolates. Granular PET is something is purified component you buy from Sigma Chemicals, right? Post-consumer PET refers to what we would commonly call a water bottle. So this is a ground up water bottle, and this is basically purchased from a provider. And you can see with the post-consumer PET that also the consortium containing all 5 isolates grew faster, steeper slope here, and a higher yield than the individual isolates. And this is grown in a carbon-free medium, meaning that it has all the essential elements for growing the bacteria, magnesium, nitrate, and so forth, but there's no carbon source. The only carbon source in that medium is the plastic. So we're forcing them to eat the plastic. So this was really kind of the first hint, or one of the major first pieces of evidence that they grow synergistically or cooperatively in culture to degrade the plastic. These series of graphs is simply to show you that the majority of the growth is found on the surface of the plastic. So it's known in the literature for a while that the bacteria have to form these biofilms on the surface. So this is really interesting because plastic is, of course, very hydrophobic, and the enzymes that degrade it have to really get into the hydrophilic interface on that surface. And so they have to form these biofilms. And so part of her thesis, Morgan took these pictures of the bacteria on the surface of the plastic, and you can see these cute little Pseudomonas species here. And this goopy stuff is actually polysaccharide. So a biofilm is composed of the bacteria with the polysaccharide over the top of it, and it creates this hydrophobic sort of interface that allow the enzyme to degrade the plastic. And so here on granular PET and the post-consumer PET water bottles, the majority for all of the isolates we see that the majority of the bacterial growth is actually on the surface. And so that was consistent with the existing literature. And also in terms of the bacterial structures, you see these projections, and these structures are called pili, or little hair-like projections that are also very hydrophobic, and they help the bacterium stick to the surface of the plastic. And as you might guess, they're also essential for biofilm formation. So I'm going to go through this slide fairly quickly to just tell you a little bit about the evidence, because I want to move on to our unpublished data. So there's a lot of different techniques. This is a biochemical technique, or Fourier transform infrared spectroscopy, known as FTIR. And all it does is it is able to detect functional groups on the surface of the plastic. So for example, if I'm going to cleave this ester bond here, I would expect to have a hydroxyl group appear, and I should be able to detect that by this infrared spectroscopy technique. And that is the case. Actually, there's a small hydroxyl peak down here that we can detect. The ester peaks go down as the ester bonds get cleaved. The carbonyl peaks go down, and the aliphatic peaks go up. And that's all depicted in this data here, where we can shine the infrared radiation onto the plastic and then detect the functional groups. That's pretty good evidence, biochemical evidence, that the bacteria are degrading the plastic. And also you'll see here the sort of the pathway that was previously worked out by other researchers. The PET plastic is degraded into BHET and MHET, and then TPA, which is going to be a major focus, as well as ethylene glycol. So to make new PET plastic, what people do is they take this monomer TPA, terephthalic acid, and ethylene glycol, and they polymerize them together to make new PET. So the degradation pathway goes this way, but to make new PET, you have to go this way. And so when I start talking about recycling, this is the molecule that I'm going to focus on. We also got some more electron microscopy images that you can see here, where this is untreated plastic. And then we basically take a mild detergent to try to remove the bacteria, but they're really tightly stuck to the plastic. And so here are the Pseudomonas, the little guys, and the Bacillus species, which are these long rods connected together, or streptobacilli. And if you then strip it away using a protein digest, you can actually see the holes in the plastic where the bacteria have been degrading the plastic. And you see then the sort of dead bacillus over here, which looks like sort of like a trough. So we had to really treat it harshly to be able to get to that point. But there's also more evidence, in this case, biophysical evidence, that the plastic is being degraded. This was published in the microbial resource announcement, where our student Cam weighed the plastic and found that, in fact, if you use the full consortium versus the other combinations of strains, that 3% weight loss was occurring, and it was greater over 6 weeks than any of the individual consortia or isolates. So again, it's another piece of evidence that they're acting cooperatively. So how are the bacteria acting synergistically, or working together, or cooperating? I also like this slide because we as scientists need to work together, work synergistically, and cooperate. So the bacteria are teaching us. Yes, I would be happy to take questions along the way. If you have questions, please feel free to put them in the chat. So I think the short answer to this question is about cross-feeding. So basically, the bacteria can only eat or degrade a certain compounds, and the other is going to be able to eat those that they cannot eat. So we know, and I'll show you evidence, that we know that the Bacillus species are very good at degrading the BHET, whereas the Pseudomonas are not. But the Pseudomonas can degrade the TPA, and the Bacillus species don't have the genes to be able to do that. They can't take it up, they can't digest it, right? So they're working together. There's an older piece of data, which I have put the reference for here, where a Japanese researchers had shown that in wastewater, that actually there's a bacterial mixed culture that degrades terephthalic acid or terephthalate. And you guessed it, the species are Pseudomonas and Bacillus, right? So it's also evidence to say that they're working together. Okay, so I have a question about, can you use other plastics, alternative to packaged water than other plastic water bottles, some readily biodegradable stuff? I think the short answer to that is yes. I think that the problem is the plasticizers. What do you put in there? Plastic is still plastic, no matter whether the material is acquired from petroleum or biologically. And some people are working at putting in ways to polymerize the material so it is more degradable. But that has been a very difficult task. So this is the evidence to show you that the Bacillus species degrade the BHET. So I've highlighted in red that point where we just feed, what my student Cam did was she took the cell lysates, basically the bacterial enzymes, and put them on the BHET. And then she monitored by NMR the presence of TPA, and really only 9.1 and 13.1 of the Bacillus species were able to cleave this molecule. And then we also see that there's synergistic growth on BHET, which does make sense. You can see here the steeper growth curve for the Bacillus species, or the consortium rather, versus the other individual isolates. In terms of TPA degradation, then only the Pseudomonas can degrade TPA. The Bacillus species does not have the genes such as those encoding this enzyme or the uptake system. Both of the strains, Pseudomonas and the Bacillus, contain the genes to be able to degrade ethylene glycol. So it's a small little alcohol that can be taken up by both of them. And so they eagerly eat this. So as I said, really the conclusion is that they're able to degrade the ethylene glycol together, but they can't degrade the other molecules. We've done quite a bit of work on trying to figure out what the initial PETase is, if you will. The PETase that was described by the Japanese group, we have some enzymes that are similar to that, but we don't really know exactly what the initial enzyme is. We've done some structural work to figure out that initial enzyme, but we don't really know that yet. Okay, so I have a couple questions here. I'm going to go ahead and take them. How much time do Bacillus and Pseudomonas take to degrade these molecules? It's very slow over the course of weeks. So far, we've shown with water bottles that we can, or plastic, we can degrade over 6 weeks, about 3%. We did another study where we only had a 2.5% over 6 months with water bottles. So it's quite slow. And we like to try to make it faster, for sure. And I'll tell you a little bit about that in my upcoming slides. Is there any degradation rate difference between microplastics and other plastics? That is a wonderful question. So a big problem, as you rightly point out, is microplastics and nanoplastics in our environment that get into the food chain and get into our own bodies. I have a thesis student this year who's looking at whether or not the bacteria can degrade nanoplastics or microplastics, and I hope we can find that they do. That is actually a very huge problem. So we've, as I mentioned, sequenced the genomes. There's a lot of interesting things there. We're just starting to get into it. There's a lot of horizontally transferred genetic information or genetic information that they've acquired from the environment. As it turns out, one of the Pseudomonas strains is more similar to the Bacillus than the other Pseudomonas strains, which is kind of crazy in terms of genetic diversity. But we're looking more into that. We know that they degrade a bunch of different type of fatty acids and polyester. And so they have a wide variety of activities. We're wondering if they can degrade additional plastics besides PET, and there's some evidence to suggest that is the case. We know that the consortium contains a large number of enzymes, alpha beta hydrolases, and we're looking into the activities or potential activities of those. You'll notice there's a polyurethanase. So we think that they can also degrade polyurethane. We know that they can degrade plastic bags. One of my students looked at that last year. Another genetic technique, which is used fairly commonly, is RNA sequencing analysis. So we're looking at the genes that are expressed during the times when they're grown on the plastics versus on a more normal carbon source, like an amino acid. And so what we want to do is look at differential expression, or what's the difference between those two conditions, right? And we've got some really interesting stuff that's going on with that. There's a lot of signaling. Obviously, they're trying to figure out what's out there in their environment to eat. There's a lot of energy and transport genes, stress response. We've got them on this plastic. Nobody likes to eat plastic. They don't like to eat plastic either. And so flagellar genes that are involved in biofilm formation. One really fascinating thing is the abundance of sporulation genes in Bacillus. Why are they sporulating? They're growing. Why are they sporulating too? Or that's an adaptation to nutrient deprivation. We think, we don't really know what's happening with that. There's a little hint in the literature, and I can tell you more at the end if you'd like to ask me about that. In the last part of my talk, I'd like to tell you our approach to biorecycling. So how might we do this? As I mentioned, PET is degraded into BHET, MHET, and then into TPA and ethylene glycol. TPA and ethylene glycol are used to combine to make new PET plastic. And what if we could harvest the TPA in a clean way using our bacteria, get them to grow faster, right? And to knock out the TPA uptake system. So we know we identified in the genome this gene PCAK in the Pseudomonas that we think was necessary for TPA uptake. And we know the Bacillus species cannot degrade the TPA, so it can just be left out there in solution. And here's the evidence that we've collected on this. And in fact, it worked. This was really exciting results because TPA is not very soluble in water, and you can see the TPA in this tube here. So we inoculated this tube with the wild type bacterium. We inoculated this tube with the strain that was deleted, had a PCAK deletion, so that gene is gone. And you can see after 2 weeks that the TPA is still largely present here. It's absent here. It's been metabolized. If you count the number of bacteria per milliliter, you can see in the wild type, there's 10 to the 7th bacteria here that have grown. And in this tube, there's only 10 per milliliter, basically nothing, except what we initially put in there. If you analyze it by high performance liquid chromatography, we put in 12 millimolar TPA here. In the delta PC, in the mutant tube, it's all still there. And in the wild type, it's about, well, 60% degraded or so. So that's kind of a proof of concept that you can actually degrade the plastic and harvest a TPA because it's still available. So where are we? I'm not going to go through this whole table, but I like to make a couple of points. So the question was, how fast does it happen? It needs to go a lot faster. If you have any ideas, please send them my way. So I shared with you that our water bottles can be graded at about 2.5% over a 6-month period at 40 degrees Celsius. So most of the data that's been collected in this area has been used on PET film or amorphous PET. That is not what we see as water bottles, which is more crystalline. So even though they can degrade this, this is not a water bottle. Post-consumer PET can be depolymerized with this leaf cutter cutinase quite readily over a short period of time at high temperature. But in the information here, I placed down here that this requires 3 milligrams of enzyme per gram of PET plastic. That's a lot of protein. If you're going to degrade metric tons of protein, of plastic, that's a lot of protein. So that's going to be quite costly. In this particular study, they were using a carboxyl esterase on water bottles, and even after a couple of days, there's very low degradation. So that's fairly consistent with what we see. Our approach then, as I mentioned, is to use the whole organism to be able to degrade the plastic. We've done some work to try to accelerate biodegradation. So we know that the bacteria degrade better at pH 8, at higher temperature. We can use solvents. Actually, ethanol acts to help increase the biofilm formation, increase the cleavage of the PNP butyrate, which is a sort of a surrogate for BHET, if you will. We've done grinding of the plastics, acid-base oxidation pretreatments, and repeat inoculations. Here's another technique that I'll share with you that is really powerful. So we can measure the biofilms in this microtiter plate assay, so this 96 well plate, which can go directly into a reader. And so basically what you do is you put the bacteria into the little well here. Then what you do is allow them to grow over days or weeks. You wash out the non-biofilm forming bacteria. Then you stain it with this purple stain called crystal violet. You wash out the excess stain, and then you put in alcohol, methanol usually, to solubilize the stain, and then you can measure it spectrophotometrically. So it's a nice way to understand biofilm formation and to quantify it. And so my student Helen did that this summer, and species 10 and 9.2 seem to be our best biofilm formers, which is a really great assay. So I have a question here. Can enzyme formulation be used to speed up the degradation process? Yes, absolutely. So so you can do directed evolution. So people have wanted to degrade the plastic, for example, at higher temperature, and you can use directed mutations to make the enzymes more stable so that they can act at a higher temperature. There's not been a lot of work in terms of actually increasing the specific activity that I'm aware of, but you can make the enzymes more amenable to degrading the plastic. And then I'll show you another thing that we've been doing, which is more of a general approach. So we've been doing what's called adaptive laboratory evolution. So what you do is you incubate. The idea is a simple one, is you just put strong selective pressure on the bacteria to evolve in the laboratory to be able to degrade the plastic better. And so last year, my student Henry, what he did was he combined the bacteria with these strong mutagens and did repeat culturing of them on PET plastic. And you can see the little goopy things are bacterial biofilms on the plastic in this flask, and then just did it repeatedly. And so he did up to 5 different generations. And you can see on the post-consumer PET that it outperformed in terms of the amount of bacteria that were growing, those on the amorphous PET. And so this is wild type consortium, mutagenized for 3 generations and mutagenized for 5 generations. And so what he's able to do is to get the bacteria to evolve in the laboratory to be able to form better biofilms on the plastic. What is the role of saponin, Krilaha? Actually, it's a wonderful mild detergent that allows you to release the bacteria from the plastic surface and to then enumerate them or count them in some way, either spectrophotometrically or by counting them by doing serial dilutions on a plate. So saponin is also used to study the interaction of bacteria on host cells, like in culture, because it won't lyse open, say, an epithelial cell in a tissue culture plate, but it will it will lyse the tissue culture epithelial cell open and release the bacteria. The bacteria are not affected by it. So conclusion then, we've isolated these 3 Pseudomonas strains and 2 Bacillus species that act synergistically in a consortium of 5 bacteria. They were isolated by my student Morgan, petroleum polluted soils. And I have a little inkling of why that is. I can share that with you if you're interested. We have evidence that they're able to degrade PET plastic because of the similarities of petroleum products and the semi-aromatic PET molecule. We have evidence that after 6 weeks, they can degrade the granular PET by 3%. And over 6 months, the water bottle picture down there by about 2.5% reduction in weight. Interestingly, then, the biology is pretty fascinating that the consortium can grow on and degrade the PET plastic more efficiently than individual isolates or individual colonies or individual consortia. So somehow they're synergistically working together to be able to do that. And we're going to continue in understanding the biology of the system, as well as working towards the goal of biorecycling or harvesting the TPA to be able to create new plastic products in a more environmentally friendly way. And lastly, then, I will end my talk by acknowledging the many students who've worked on the project since Morgan started in 2017, continuing now through this year and on beyond. I've had a high school student who worked on part of the project as well, and then collaborators Rosa Leon-Zayas, who's at Willamette, which is close to Reed College, my wonderful research associate Sabrina, as well as funding from the National Science Foundation. And Morgan's TED Talk has gotten quite a number of listens. It's a really, she does a really wonderful job of explaining how she isolated the bacteria. And with that, I'll stop and take any more questions that you'd like to ask.
Yes, sir. Thank you so much for that enlightening webinar. I really found it fascinating how, I mean, I thought it was a very hopeful webinar, because normally you hear all the time that there's plastic pollution and, you know, plastic is non-biodegradable, and then you're like, oh my God, what's happening, right? So,
yeah.
So this was very hopeful for me, because even though the progress is small, but it's there, right? So that was very enlightening for me. So, yeah. So, right. So you took the questions in the chat box. That's very kind of you. There are some questions, other questions that we'd like to ask you, if that's okay.
Yeah, sure.
Can I interrupt for a minute?
Yes, sure, ma'am. Sure.
Yeah. So, Dr. J, actually, I would say that it is really wonderful to know that Bacillus and Pseudomonas are actually on their role. They degrade a lot of things. I'm really amazed. They degrade many pesticides. They have capability of doing a lot wonders to the environment. I'm really happy to know that Bacillus and Pseudomonas are active over here also. Actually,
absolutely.
Yeah. Like my supervisor used to work on HCH degradation. There also we used to learn about Bacillus, Pseudomonas, Fingomonas and all. But in plastics also,
yeah,
it's really nice to know. Yeah.
Yeah.
Thank you.
The genomes have an amazing number of enzymes. There are about 7,000 genes in each of those bacteria, exactly, which is a huge number of genes. So they're really, we did this experiment using, there's a company called Biolog, and they provide these plates to be able to understand the different carbon sources the bacteria can use. It's used to characterize soil bacteria. In these plates is about 30 different carbon sources. You guessed it, our bugs could eat every one of them.
The thing is, Bacillus and Pseudomonas, they can degrade any pollutant present in water, agriculture, plastics also, pesticides present in soil. I mean, wonderful organisms, I must say.
Yeah, for sure.
Thank you.
Right. So Yeah, thank you for that insight. I didn't know that, so thank you. So right, the first question. So we did a little background study. So we came to know that you studied music as a minor in your college life. So I was wondering what made you decide to choose music, like with microbiology. That's a very fascinating combination.
Thank you for that question. As it turns out, when I was studying music, I realized that scientists, many of them, are musicians too. And I think it's because of the order and the structure and the numbers and the chords and the minor thirds and the major thirds and all that, right? So it appeals to, I think, a scientific mind. And I think it's a common thing. And so I started taking classes in music because I was involved in music when I was young. And I just said, hey, I can minor in this. And I just kept going.
That's really good. That's really amazing. That's something I want to learn in my future as well, music properly.
Mm-hmm.
Vipashadi, can you take the second question?
Thank you, Ranjini. Hi, sir. It's an honor to be sharing the screen with you. And the talk was very, very enlightening, as Ranjini said. So I had a question too. So can there be such a thing as sustainable plastic in the future, since some bacteria are able to degrade plastics, like you said?
I'd like to think so. I think that per the other question about making plastics so they're more biodegradable, I think a whole area that we've not approached is policy, governmental policy or regulations that make the plastic producers be more responsible. So take the phthalates and the other plasticizers out of the plastics and make them so they're more biodegradable or at least less harmful, and have it more standard so we can purify the components. Because even if you do chemical recycling, there are so many pollutants that most of the material is just waste. So let's fix that. I think that across the globe, we need to advocate for more responsible plastic production.
Yes, definitely. So thank you for that. And moving on to the third question. So with the knowledge that plastics can be degraded to some extent, boom plastic production and consumption, like would people think that, this is degradable as well, we'll use it. So like, what's your opinion?
I was aware of a British company that was trying to produce a product where you could place a water bottle into a landfill and in 2 years it would be gone and biodegraded. And one of the arguments against that was, just as you share, then, I'll just throw it away because it's going to degrade anyway. And some of the argument was that people would just be irresponsible with handling the materials. So yeah, I don't know. I think I'm not sure about that. What do you think about that?
Yeah, we would. Next policy changes. Yep.
Yeah.
So there is one question in the chat box, actually. I'll just dictate it out now. Could this kind of extensive research on various strains of bacteria create a risk where such bacteria develop strains that are harmful to humans and or other organisms?
Yeah, that's a really interesting question. I had I think the answer to that is no. These are soil bacteria. We live, all of us, amongst bacteria every day, right? There's 10 to the 30th bacteria on the planet or whatever. We evolved together with bacteria. And so I think, not to minimize it, for example, those of us who are immunocompromised are susceptible to Pseudomonas infections, right? So people who have cystic fibrosis or burn victims. So it is the case that, of course, they can be harmful under certain circumstances. But these bacteria are out there. They're evolving already. There was an interesting preprint. I'm not sure if it's been published yet, on showing that bacteria in the oceans are actually evolving to degrade plastics. Why not? Of course, any carbon molecule out in the environment is going to be able to be degraded by bacteria. It's a matter of time and how fast it goes or how slow it goes, right? Yeah.
Yes.
Also, horizontal gene transfer may contribute to this in future.
Yeah.
Maybe some other bacteria would like to pick these genes.
Absolutely. I mean, that's why it really, in a fascinating way, the bacteria that we isolated, the Pseudomonas is more similar to the Bacillus species. That has to be horizontal transfer, right? We haven't been able, we haven't had time to do the analysis yet, but Matsi, you're absolutely right. It's got to be horizontal gene transfer because those genes are being passed along and they're being amplified in the community because those are the genes that are necessary for the different types of bacteria to degrade the plastic, right?
After all, it's all about evolution.
Yeah.
That's how species evolve.
Yeah, for sure.
Yeah. And can there be any enzymatic formulation which can be directly added onto these plastic dumped areas where degradation can act faster? Rather than having microbes, we can have extractions from them, like purified enzymes. Will that be a solution? Can it propose a solution for such a huge chunk of plastic?
Yes, I mean, I think that that's possible. I have, in all sincerity, I've never thought that we would take our bacteria and take them out to the gyres in the ocean or to landfills or places where plastics exist and release them, right? I've never thought that I would do that. But sure, if you could isolate enough enzymes to squirt it on to plastic waste dumps, you could try to get it to be degraded. I think the hazard...
Yes, because for certain pesticides, this is a practice which is done or being tested, that you can like get concentrated enzymes, and if you put them directly, so they will have lesser negative effects in the environment, because even if we add too much of bacteria, that may disturb the ecosystem. So that is why the concentrated enzyme, their formulation can be added.
Mm-hmm. Yeah, I think, yeah, that's a good alternative to a whole organism, right? Yeah.
Yeah. So, okay. So thank you so much for taking all those questions. I'd like to conclude the question answers, but conclude the question answers.